Hello, my name is James and welcome to my YouTube channel. I first wish to mention that I in no way endorse myself or wish to represent myself as being an authority on the subject of the species of tropical spiny lobster known as the Panulirus ornatus. Rather, I am merely a novice student of the subject myself. I am presently living on a small island here in the Philippines, overseeing the construction of a prototype lobster microhatchery facility, which I designed a few years back. And for the last 9 years, both lobster hatchery, mariculture, and lobster grow-out aquaculture has been both a passion and a personal obsession of mine. I have devoted countless hours on the internet trying to seek any useful information pertaining to both lobster aquaculture and mariculture, so I can fully understand and appreciate both the difficulty and frustration one may encounter when trying to research the subject.
When researching the lobster aquacultural industry, you quickly discover the topic actually encompasses a myriad of different subjects, and the information is scattered throughout hundreds of PDF research documents, e-books, textbooks, websites, and internet videos. But these sources also seem to be conflicting and confusing with large gaps of missing needed information. I know that even after 9 years of personal research on the subject, it still seems like I'm still piecing together a large complex jigsaw puzzle without many of the key pieces. These gaps are because there is still much about the Panulirus ornatus that the researchers still just do not fully understand. This is because much of their early developmental stages actually occur thousands of kilometers from the shorelines, out in that mysterious deep blue regions of our vast oceans. Currently, researchers actually know very little about their true natural diet during their long larval stages, or which are their desired food preferences as they transition through each developmental stage. Such information is vital to help ensure the adequate nutritional diet is provided through each of their growth cycles. This is because in nature they are drifting amongst the phytoplankton and zooplankton layer of the ocean depths of 30 meters, somewhere out in that deep blue. New information often comes from brief glimpses and mere happenstance, such as a photo taken by a diver or a specimen captured in a fisherman's net.
Researchers desperately wish to understand the species, so they can try to finally simulate what nature is accomplishing in the huge larval nurseries of the wild. But presently, researchers can only really observe from within close proximity to coastlines, far from their larval existence out beyond the continental shelves. One also has to understand that if there is new information being derived from recent research studies, that it is more probable that little will be openly shared on the internet. I personally believe this is primarily due to language barriers, not being translated into English and shared, or perhaps due to policies involved in corporate, government, and university-sponsored research, which gets quickly bogged down in the politics of non-disclosure clauses and proprietary intellectual property claims. This is certainly understandable, especially with feed and formulation studies, whose sponsors and investors seek potential financial rewards in return for their investments. They want to capitalize on the huge financial potential through the creation of the elusive holy grail of the lobster aquacultural industry, which is to be the first to market a viable formulated dry mix equivalent to a lobster version of puppy chow.
For 9 years I've collected useful information, facts, and figures on the Panulirus ornatus species. These I organized into a 3-part video presentation designed to present this information focusing on lobster grow-out aquaculture and the lobster grow-out industry. I will discuss in depth the complex biology, physiology, and life cycle of the species as they directly relate to their lengthy developmental stages. This three-part video series will primarily focus on the aquacultural grow-out practices necessary to successfully raise early-stage Puri to marketable 500-gram subadult and 1,000-gram adult-size individuals. This discussion encompasses in-depth topics on primary water management and controls, optimum water levels and conditions, proper husbandry practices, and general care and net management. The intent of the video series is to provide the fundamental knowledge needed to best choose the proper location to construct a lobster grow-out aquacultural operation while avoiding common mistakes, counterproductive practices, often unseen pitfalls, which can seriously hamper the success and profitability of a grow-out aquacultural operation. I will also be discussing the lucrative lobster live capture collection industry and many of the various methods, equipment, and techniques presently being successfully deployed by the Pueruli and Juvenile seedstock fishermen within this growing industry. Related topics covered will include how to properly handle live capture, Pueruli, and Juvenile seedstock, how to properly house and care for Pueruli and Juvenile seedstock, and how to presently prepare for and successfully transport live Pueruli and Juvenile seed stock to the lobster grow-out operations. If you find this three-part video series to be both informative and entertaining and wish to watch upcoming videos in this video series on my YouTube channel, then please feel free to subscribe to this channel. This will ensure that you will be informed when new videos of this series are available for viewing. Also, please feel free to like and share my videos and leave a comment if you feel so inclined. If any of this information is in error, I will be happy to make concession as new information becomes available. It will be upgraded onto the video. Thank you. So now that this introduction is concluded, let's begin seriously talking lobster.
In the lobster grow-out aquacultural industries of India, Indonesia, Malaysia, Philippines, Thailand, and Vietnam, they are greatly dependent on the Panulirus species lobster, especially the subspecies known as the Panulirus, which yields a large portion of their annual production. Here in the Philippines, there are 8 species of Panulirus lobsters, which are found in the waters around the islands of the Philippines, and these species fuel the local lobster grow-out industry. These species are the Panulirus homarus, or scalloped or cancer homarus, Panulirus japonicus, or Japanese spiny, Panulirus long-ipes, or long-legged spiny, Panulirus ornatus, or tiger ornate spiny, or tropical rock, the Panulirus penicillatus, or gray cray, the Panulirus polyphagus, or mud spiny, the Panulirus simpsoni, or Chinese spiny, and the Panulirus versicolor, or bamboo painted spiny. These 8 species of Panulirus fuel the Philippines grow-out aquacultural industry. The Panulirus species which I primarily focus most of my attention is the Panulirus ornatus, which is commonly called the ornate spiny lobster, ornate tropical rock lobster, ornate rock lobster, tropical rock lobster, pearl lobster, or as they are commonly referred to here in the Philippines, tiger lobster. There are 22 species of Panulirus within the genus Panuliriid, and they can be easily distinguished by their long flagella on their first antennae, a subcylindrical carapace with horns over the eyes and long antennae. These first 2 antennae can grow up to 2 feet, 61 centimeters long. They are used to sniff out its prey. The Panulirus ornatus is known as stridentes due to its sound-generating stratilating apparatus at the base of the antennae. If threatened by prey or aggressive lobster, they rub their antennae against their head, which makes a loud grinding sound. The ornatus is known for its distinctive 7-color exoskeleton. These colors are black, blue, brown, green, orange, white, and yellow. The Panulirus ornatus have rather poor eyesight but possess highly developed senses of both taste and smell. They are poikilotherms, meaning cool-blooded. The temperature of their environment directly affects their food intake, growth rates, molting increments, intermolt periods, egg incubation times, and their mortality rates. The Panulirus ornatus is a reef-dwelling, 10-legged crustacean, which is closely related to both shrimp and crab. They are found in the tropical waters of Australia, Fiji, Indo-Western Pacific Ocean, Indonesia, Japan, Malaysia, New Caledonia, Pohnpei, New Guinea, Philippine Sea, Red Sea, Solomon Islands, South China Sea, Southeast Africa, Southeast Asia, Suez Canal, and Vietnam. They are termed a continental species because they are common in moderately turbid waters and rocky areas on broad continental shelf areas of the larger islands and continents. Generally at water depths ranging from 9 meters to 500 meters with a terrestrial influence. These turbid waters provide moderate wave energy action, especially along coral reefs and back reefs, where prolific coral cover provides sufficient sheltering and support diverse food sources. But the Panulirus ornatus can also be found in sheltered waters of lagoons and high turbid silted rubble areas near river mouths and in mangroves. They generally forage over relatively flat rocky seabed rather than on high relief coral structures. In the wild, males are believed to live upwards of 35 years, whereas females live upwards of 50 years. They can achieve a weight of 7 to 10 kilograms. Here in the waters of the Philippines, we naturally have 3 of the most prized species of Panulirus. These are the Panulirus ornatus, tiger, the Panulirus versicolor, bamboo, and the Panulirus hormeris, scalloped. Of these 3 species of Panulirus, it is the Panulirus ornatus which is considered the most prized in the lucrative Asian lobster markets. The Panulirus ornatus traditionally brings one of the highest sales prices for delivered live lobsters. There are several attributes particular to the Panulirus ornatus which contributes to this popularity and high demand in the markets of China, Hong Kong, Japan, Singapore, and South Korea. They are prized for their creamy textured, rich, pearl white color flesh and the natural sweet taste of the meat which is used in sashimi and in sushi type dishes. The Panulirus ornatus also yields 43% meat of total body weight, compared to only 23% meat yielded of total body weight found in cold water clawed species of Europe, Canada, and the United States. It should be noted that the female yields the largest tail by weight and size, and the flesh yielded by the female has a sweeter flavor than that of the males. The second attribute is their 7-color porcelain-like exoskeleton, because this shell makes an attractive center display for restaurant presentations, celebratory banquets, and wedding receptions. It is considered a statement of luxury and prestige, and when the lobster is cooked, the lobster shell turns a deep red, which is considered a very lucky color in China, Hong Kong, and Singapore. The 2014 off-season Asian market wholesale price for a 1 kilogram plus size Panulirus ornatus reached $120 per kilogram during the Chinese New Year, and consistently yields prices which average $65 to $75 year-round. There are also favorable attributes particular to the Panulirus ornatus, which makes the species the center of attention of the lobster grow-out aquacultural industry. The ability of the Panulirus ornatus to both survive and thrive in such a wide range of diverse habitats, such as close proximity to coastal shorelines, among mangrove forests, and in areas with a minimum water depth of 1 meter, sets them apart from other lobster species. This allows them to be successfully raised in the captive environment of shallow shrimp ponds, nets, cages, floating platforms, and heavy sea cages, which makes lobster grow-out aquaculture an excellent small business venture for coastal Filipinos, especially considering the over 14,000 kilometers of shorelines. Growers can raise a 3-gram Puri to 1/2 kilo, 500 gram, in only 8 to 12 months. A 3-gram Puri to 1 kilo, 1,000 gram, in only 14 to 18 months, and a 3-gram Puri to 1.5 kilogram, 1,500 gram, in only 18 to 24 months. Buyers purchase lobsters from the growers at 3 different minimum size requirements, bringing 3 different buyer price ranges. This allows growers more diversity and ability to specialize their grow-out operations. This fast growth rate also allows providers an opportunity for grow-out operations to have faster stock turnover ratios and lower production overhead cost. The higher market cost and high demand for the Panulirus ornatus provides the greatest potential for high profit yields. Common terms relating to Panulirus ornatus. Kingdom of Animalia. Phylum of Arthropoda. Class of Malacostraca. Order of Decapoda. Superfamily of Palanuroidea. Family of Palanuridae. Genus of Panulirus. Panulirus. 22 species of lobster within the taxidermic family of Palanuridae. Tropical or subtropical shallow water modem lobsters. Panulirus ornatus. Specific species with 7 distinctive color exoskeleton. Colors are black, blue, brown, green, orange, white, and yellow. Nepulae. Singular term. First larval stage of development upon leaving the egg. Nepliosoma. Plural term. First larval stage of development upon leaving the egg. Nepulus. Plural term. The first larval stage upon leaving the egg of many crustaceans. Phylosoma. Singular term. Early stage lobster larvae. Greek meaning leaf body. Phylosomata. Plural term. Early stage lobster larvae. Greek meaning leaf body. Puerulus. Singular term. Post-larval stage of spiny lobsters. Puri. Plural term. Post-larval stage of spiny lobster. Glossary. Benthic. Relating to the bottom of a sea or lake or to organisms that live there. Carapace. Refers to the lobster's body or back. Carapace length. CL. The length of a carapace back is usually measured in millimeters. Caryopsis. The ability of an organism to conceal itself from predator by having a color pattern and shape that allows it to blend into the surrounding environment. Instar. Refers to the intermolt period between 2 successive ecdyses, whereas stage denotes 1 or more specific morphological characteristics unique to that phylosoma. Thus a stage can include 1 or more instars. Invertebrates. Animals that do not have a backbone. Common examples include crabs, lobsters, snails, sea urchins, insects, worms, etc. Leicithiotropic,
meaning feeding on yolk.
non-feeding, drawing from their own stored energy reserves. Micronecton, relatively small but active swimming organisms ranging in size between plankton, less than 2 millimeters, which drift with the currents, and larger nekton, less than 10 millimeters, which have the ability to swim freely without being overly affected by currents. Nektonic, actively swimming aquatic organisms in a body of water able to move independently of water currents. Oligotrophic, lacking in plant nutrients and having a large amount of dissolved oxygen throughout, such as deep ocean or lake environments. Ovipositioned, to lay deposit eggs with the use of an egg-laying tubular structure at the end of the abdomen. Pelagic, living or occurring in the upper waters of open sea. Planktonic, drifting in water. Peleopods, also known as swimmerets. They are found on the underside of the lobster tail. They are used for swimming, burrowing, ventilating, and carrying eggs in the female. The first pair is different between male and females and is used to identify its sex. Poikiliotherms, cool-blooded. Growth rate is directly correlated with the temperature of their environment. Substrate refers to the material covering the surface. For example, sand is a substrate covering part of the ocean bottom. Mud is another. In the wild, maturing individuals have been observed walking distances of 600 kilometers to reach breeding grounds during their mating spawning season. Compared to other species of Panulirus lobsters, the Panulirus ornatus are active for only a few months yearly. Their mating spawning season is during the summer season, when food is far more abundant. The photoperiod ratio of the sun and moon cycles are 14 to 14.5 hours sun and 9.5 to 10 hours dark, and the water temperatures are at their warmest 29 centigrade. In the wild, female Panulirus ornatus will slightly outnumber the male population. The female Panulirus ornatus can reach breeding size much quicker than other Panulirus species, being capable of reaching sexual maturity and breeding in only 2 years from a hatchling size. The females are generally sexually mature once she reaches a 90 millimeter carapace length, CL, though she may start carrying eggs at a 60 millimeter carapace length, CL. Females are substantially heavier weight than males for the same length and size. Also, females with eggs are heavier than females without eggs. The females can easily produce 3 or more broods within this short mating season, depending on her maturity, size, and general health. A Panulirus ornatus female can easily produce 400,000 to 1.6 million eggs per mating season. Females can spawn 3 to 5 times during a single mating season, but generally only 3 times during a mating season. Both females and males can have multiple breeding partners during a single mating season. Typically, the females molt 3 times per year. These 3 molts are referred to as the pre-reproductive molt, which precedes the beginning of her spawning cycles during the mating season. The post-reproductive molt, which actually terminates her spawning cycles at the end of the mating season. And the growth molt, if it occurs, as it's dependent on the age and general health of the female. The mating, egg exclusion, and hatching occurs during the intermolt period, when the shell is hard. During the warmest summer months and the extended daylight hours provide the natural cues which trigger the pre-reproductive molt. The female develops hairs on her peleopod setae, which is where the ovipositioned eggs will be adhered and will be incubated by the female until hatching. The female will begin to accumulate the needed energy reserves required for reproduction. During these 3 months, her reproductive system fully develops, and she also begins to build up her natural energy reserves, which are paramount before her eggs are laid. A female typically experiences a 7% decline of total body mass after each spawning event, and a female can easily spawn 3 times in a single mating season. So a female can easily lose a quarter of her total body mass after 3 consecutive spawnings. This is why having an adequate diet is so important, as it significantly allows her to build up her energy reserves. This will also affect the quality of the embryos she produces, as well as her general health. Depending on her age and general health, a female Panulirus ornatus can lay well over 1.6 million eggs over 3 consecutive spawning cycles within only a 4-month mating season. It is believed that the male may be attracted to the female by a possible sex pheromone secreted by the female through her urine. Lobsters actually excrete urine through the openings of their face, located at the base of their second antennae. Both the male and female have their bladder located below the brain and 2 small sac reservoirs of urine located just under their eye sockets. These are commonly referred to as green glands. The organs that excrete waste include a sac that is linked to the lobster's bladder. These 2 sac reservoirs directly connect 2 small nozzle-type duct appendages, which allows them to squirt urine up to 7 times their body length out in front of them, which they use as a defensive measure. Females use this to squirt urine and possible sex pheromone in front of the males during their mating rituals to make them less aggressive and more receptive. Females have been observed actually squirting urine at the shelter entrance of aggressive males and repeat this over several days. She may make daily visits to the male's shelter and quickly flick her antennuals inside to smell if he is there. Then she quickly squirts a stream of urine in his face and then quickly retreats. A more aggressive male may lunge at an advancing female, but eventually the male will become more receptive and will also squirt his urine at the female. Now that the female has approached the potential mate and found the male receptive to her advances, she will then pre-reproductive molt, shedding her old exoskeleton. During this time, she will be vulnerable. The male partner helps guard her while she has time for her new exoskeleton to properly harden. Mating, egg extrusion, and hatching all takes place during the intramolt period, following the pre-reproductive molt, once her exoskeleton is hard. The male will mate with intermolt females that have developing ovaries. He will follow the female sometimes for many hours, touching her continuously with his antennuals and the first 3 pairs of walking legs. The intensity of this interaction increases, and eventually the male will rush head-on at the female in an attempt to grab, lift, and position the female into the mating position. This attempt may be unsuccessful and require several attempts before being successful. The female may take up a vertical position or even lie on her back while oviposition occurs. The male will lift the female into contact with the sternum for 5 to 8 seconds, where he deposits an acellular mass containing tubular spermatospores, which he releases from his gonospores, which are paired penile projections at the base of the 5th legs, onto the sternum, located on the 4th and 5th sternal plates of the female. These spermatospores may remain there for weeks or even months. I even read once where a female could carry the sperm for up to 2 years before deciding to fertilize her eggs. Now, if this is true or not, I really do not know for sure, but I have seen it repeatedly cited that the spermatospores deposited by the male are actually shared between two separate spawning events during a single mating season. Once the female is ready, she will extrude several 100,000 eggs from the paired ginospores located at the base of her third walking legs in a temporary breeding chamber she forms by tightly curling the abdomen over her sternum, and at the same time she will release eggs from her ginospores at the base of her third periopods. These eggs are drawn into the chamber by a current she creates by the beating abdominal appendages, peliopods. The female then releases the non-molten sperm by scraping open the spermatospores with special chelae located at the ductal of the fifth walking legs. The sperm are motile, can swim in water and swim about under her closed tail while she extrudes sticky eggs into the hairs, setae, under her tail. The fertilized eggs then become attached to the long ovigerous setae of the endopodidae of the variomose peliopods for about 6 to 10 weeks, depending on water temperature. Hotter means faster growth and shorter gestation. There they will progress from orange in color as they are full of yolk to dark gray when the yolk is used up and the lobster larvae, Phylosoma, can be seen through the egg case. The eggs are then carried under the tail of the female for about a month. Then she will migrate to the deeper water or to an area bordering oceanic water mass where currents are stronger. When the time is right, generally during a full moon cycle, the female will stimulate and release her tiny Phylosoma larvae by rapidly flexing her raised abdomen, and the currents will directly carry the larvae into the oceanic environment. The developmental stages of the Panulirus ornatus. There are 11 distinctive stages of development: egg, embryo, naplosoma, Phylosoma, planktonic Puerulus, Puri, agile juvenile, post-agile juvenile, late juvenile, subadult, and adult. Within those 11 stages of development, there are 11 embryonic phases, 11 larvae phases, 4 Puri phases, and 4 juvenile phases. The adult stage is the last developmental phase, but they will continue to grow in size and maturity. Instar, molt, TW, total body weight, and CL, carapace length as developmental stage markers. At the end of a developmental phase or stage, there is a metamorphosis involving the shedding of their old exoskeleton. This molting of the old exoskeleton during the metamorphosis is referred to as an instar. After the egg hatches, the individual hatchling is called a nepulus, and after 2 to 3 hours, the nepulus instar, molting into a stage 1 Phylosomata larvae. The Phylosomata will then develop through 11 distinctive larvae stages, involving between 21 and 24 instars. When the individual stage 11 Phylosomata completes the final larvae instar, they are now planktonic Puerulus. There are 4 distinctive Puerulus developmental stages: planktonic, clear, H-phase, and pigment. But there is only one instar upon the completion of the pigment Puerulus stage, when the individual molts into an agile juvenile. There are 4 distinctive juvenile developmental stages: agile, post-agile, late juvenile, and subadult. But these developmental stages are determined by the individual TW, total weight, or CL, carapace length. This is also true with identifying transition to the adult and mature adult stages. Egg stage. Once fertilized eggs of an embryo stage begins, the eggs will incubate between 21 to 36 days. This is dependent on the water temperature and salinity level. Embryo stage. During this incubation period, the embryos go through 11 distinctive embryonic developmental stages. The lunar cycle triggers the hatching of eggs. Once the embryos of the viable fertilized eggs have been fully developed and are ready to hatch, they will begin to hatch at night and often will hatch over 2 consecutive nights. It is during this time the female will raise her tail and gently fan, which gently releases the larval hatchlings into the currents. These earliest stage hatchlings are called neplosoma. The Panulirus ornatus actually has the shortest larval cycle of any of the spiny lobster species, which can be 90 to 120 days, depending on diet and water temperature. Neplosoma stage. These newly hatched larvae are now in the first larval developmental stage, called either napulus, singular, or neplosoma, plural. These neplosoma are less than 2 millimeters total body length in size and swim by beating their first antennae. They instinctively swim towards the water surface, being naturally attracted to moonlight or other light sources, much like moths do towards a flame or light source. This is a true non-feeding, non-cannibalistic developmental phase. The neplosoma larval stage actually lasts only 3 to 12 hours before they instar, but most written sources cite 3 to 4 hours. I can only assume the difference is dependent on the water temperature. Once these neplosoma instar molt, they molt into stage 1 phylosomata. Phylosoma stage. After these neplosoma instar, they are post-neplosoma and have molted to stage 1 phylosoma, which has a 1.56 to 1.66 millimeter total body length. They are referred to as either phylosoma, singular term, or phylosomata, plural term. These stage 2 phylosoma are a transparent, flattened, leaf-like form with short, simple antennae and use the feathery expodites of the walking legs for swimming. The phylosoma stage consists of 11 distinctive developmental stages and progress through 18 to 24 instars, molts. As these larvae continue to develop and grow, they will often instar molt multiple times during a single larvae stage. During each of these 11 stages, they will continue to grow in size and physiological development, but will still maintain their transparent, leaf-like physiology. Their gills and fifth walking legs do not develop until later phylosoma developmental stages. The tail becomes progressively more developed, and the main second antennae, which are shorter than the antennules in earlier stages, become relatively much longer. The phylosoma developmental stage is their longest pre-adult developmental phase. In the wild, the planktonic life of phylosomata is generally between 4.5 months and 9 months, but in the controlled environment of a hatchery, this time frame has been reduced to between 4 months and 7.5 months. It is during the phylosoma developmental stages that they will go through the most morphological changes. In the wild, these planktonic phylosomata are pelagic and dispersed among the zooplankton, where they drift with the zooplankton, being aimlessly carried with the currents over distances of hundreds or even thousands of kilometers in the mysterious oligothropic environments of the ocean sea. These early developing larvae only have limited vertical mobility, which allows them to migrate with the zooplankton and micronecton biomass. Though they possess little to no horizontal mobility, this limited vertical movement allows them to ascend towards the surface during night hours and descend to depths during daylight hours, and also allows them to catch live prey among the zooplankton. The average size of a latent stage 1 phylosomata has a TBL total body length of 1.56 to 1.66 millimeters and a CL carapace length of 0.5 to 0.7 millimeter before their stage 1 instar molt. Once the same phylosomata have fully reached the size of a latent stage 11 phylosomata, Their average size will have grown to a TBL, total body length of 15.9 to 16.4 millimeter, and a CL carapace length of 11.6 to 11.8 millimeter. As the Phyllisomata transition through each of the 11 stages, their average growth per stage is 0.4 millimeter to 0.6 millimeter TBL total body length and 0.3 to 0.6 millimeter CL carapace length. The duration of each of these 11 developmental stages directly correlates with both water temperature and nutrition. Once a latent phase, stage 11, Phyllisomata instar, this molt is referred to as the penultimate instar. After the penultimate instar, they morph to the postlarvae Platonic Puri developmental stage. It needs to be noted that when latent 11, stage 10, and perhaps even stage 9 Phyllisoma are carried into areas that have a water depth which is less than 200 meters, they are natural environmental triggers which will induce Phyllisoma to penultimate instar and morph to the Platonic Puri stage of development. Also, the penultimate instar has an extremely high mortality rate. Puri stage. Once late-stage Phyllisoma successfully complete the penultimate instar, they morph to the first stage postlarval Puri, known as planktonic Puri. There are 4 distinctive Purilus developmental phases, referred to as the Stage 1 planktonic Puri, Stage 2 clear postmort Puri, Stage 3 H-phase Puri, and Stage 4 pigment Puri. During these 4 Puri phases of development, these postlarvae are lehisithiotropic, meaning that they draw on their own stored reserves and are in a non-feeding phase of development. The time frame generally needed for Puri to complete all 4 Purilus developmental stages and instar into agile juveniles is 18 to 27 days. This variation is dependent on water temperatures. At the water temperature of 28 centigrade, it takes between 20 to 27 days to complete the transition from planktonic Puri to the final Purilus instar. But at a water temperature of 31 centigrade, this time frame is reduced to only 18 to 24 days. And even though it is widely written that Puri during all 4 stages of development are strictly a non-feeding phase, I have read some feed-oriented research studies relating to the Panulirus ornatus, which cite mortalities due to the act of cannibalism. So I'm on the fence on this one until researchers make up their mind. So if you purchase Puri for your own grow-out nets and cages, I suggest that you err on caution and provide sufficient mesh shelters in your enclosures. Because if in controlled studies which house Puri together in a non-feeding phase, then there should not have been acts of cannibalism reported. Perhaps these researchers of these feed studies misidentified juveniles for Puri, as it would seem odd to be conducting feed studies on supposed non-feeding individuals. But once the non-feeding Puri fully develop through all 4 Puri developmental stages and instar molt morphing to post-Puri agile juveniles, then they would once again begin to feed. Puri are naturally solitary by nature and seek sheltering upon settlement. Planktonic Purilus stage. Stage 1 postlarval Puri phase. There is much about this nectonic Puri phase which is still a mystery for researchers. This is primarily because this is where latent stage 11 Phyllisoma morph to Platonic Puri at the edge of the shelf break. The average width of a continental shelf is 45 to 80 kilometers, but the width can be considerably more depending on the topography of the seafloor. This is where the ocean currents will carry latent planktonic stage 11 Phyllisoma over a shelf break, where the continental shelf joins the continental slope at the water depth of 200 meters. Locations where there is more of a drop-off rather than a gentle slope. This drop-off topography, with a lacking of sloping, appears to be a natural cue triggering latent stage 11 phylosoma to begin the penultimate instar molt. The penultimate instar is the last phylosomata metamorphosis and the transition to the stage 1 planktonic Panulirus. This makes sense as the waters out beyond the shelf break are natural areas with high concentrations of plankton, phytoplankton, and algae, which is both a nursery and primary food source for latent planktonic phylosoma. These areas are often associated with the formation of tropical storm fronts and cyclonic wind development. This cyclonic activity is the same with cyclones, typhoons, and hurricanes, which are primarily only distinguishable by global locations. There is a theory that tropical storm fronts, when developing over waters with minimum water depths of 200 meters, will be a natural trigger for latent stage phylosoma to enter their final instar, penultimate molt. The theory suggests that the winds generated by the storm fronts create a pressurization of the sea surface water associated with water current flows. Planktonic phylosoma and early stage larvae, Platonic Puri, are naturally found at depths of 30 to 60 meters during the nights and down to depths as low as 100 meters during daylight hours. Tropical storms generally generate two types of surface winds, known as cyclonic winds and anticyclonic winds. Both in conjunction with Eichmann's transport create the sea surface conditions referred to as divergence and convergence. Cyclonic winds applies additional pressure directly above the sea surface, which literally presses down on the sea surface, causing surface divergence. The surface divergence, being pushed by the thermocline, creates an upwelling current. Anticyclonic winds cause a decrease of surface pressure above the sea surface, causing surface convergence. The surface convergence, reacting with the thermocline, creates a downwelling. Both the cyclonic wind and anticyclonic winds of tropical storm fronts gently push stage 11 Platonic phylosoma and Platonic Puri over the shelf break as the storm fronts move forward. The shelf break is the crest of the continental slope. This is where the depth and degree of slope is important. If the slope is gradual, so will be the increase of pressure. The steeper the slope, the greater and more intense the increase of water pressure as the Platonic phylosoma pass over the shelf break. As currents carry the Platonic phylosoma over the steep continental slope and into the noritic waters of the continental shelf, it causes a sudden increase in water pressure, triggering the natural cue, inducing their final penultimate instar molt. This seems to make sense as it's substantiated by another current theory, which believes that Panulirus ornatus latent stage phylosoma, which are at least 120 days old, can actually complete their penultimate instar if water currents carry these phylosoma into areas where the water depth is less than 200 meters. Looking at both of these theories, it would seem that increases in water pressure is a natural cue that triggers the final penultimate instar molt, morphing to postlarval planktonic Puri. In the wild, it is believed that out of a single brood easily numbering between 200,000 and 800,000 individuals, less than 10% actually successfully complete the penultimate instar. This exceptionally high mortality rate is primarily due to morph death syndrome, MDS, which is caused by the inability of lobster to properly free themselves from their old exoskeleton during instar molts. Researchers believe that MDS during the penultimate instar is due to diets lacking in sufficient nutrients during their phylosoma developmental stages. It is also believed that if stage 10 and stage 9 phylosoma are carried into areas with water depth less than 200 meters, are naturally cued to penultimate instar. And these early stage phylosoma are then underdeveloped and do not yet have sufficient stored lipid reserves to successfully penultimate instar. These planktonic Puri are no longer zool larvae and have now changed in behavior, locomotion, and metabolism. The planktonic Puri phase of development is a transitional phase between the previous pelagic planktonic phase and the new nectic phase. This will be their last true swimming stage of development. They are no longer restricted to merely vertical movement and having to aimlessly be carried along the ocean currents. After the penultimate molt, they now also possess a well-adapted, strong horizontal directional swimming ability by beating their peliopods, as well as using the familiar tail-flicking escape response of adult lobsters. For the first time, they are capable of moving independent of water currents. Post-larval stage 1 planktonic Puri are transparent with a total carapace length CL of 30 to 35 mm and generally weigh less than 1 gram dry weight. The planktonic Puri stage of development is classified as a non-feeding phase, but I personally question its prevailing belief as actually being completely factual. They are naturally nocturnal and are sparsely distributed among the zooplankton and now no longer aggregate towards the surface at night. They must complete a life-and-death migratory swim across the continental shelf towards the shallow grasslands and coral zones near the shorelines. Ocean currents play a major role in the success of planktonic Puri to complete this migration. A healthy planktonic Puri can swim an average of 7 kilometers, 4.3 miles per day. Migrating planktonic Puri primarily swim with the ocean currents during the new moon, no moon, lunar cycles, when the currents are at their strongest. Swimming during the new moon lunar cycles also allows the Puri to use the cover of darkness to help evade predators. During this nocturnal migration, these Puri will rest during the daylight hours under stones or burrowed shallowly into the sand on the ocean floor. This helps to shield and conceal them from predation and allows them to rest and conserve their energy reserves. The 4 distinctive developmental stages of Puri are each non-feeding phases, so these Puri must metabolize stored lipid reserves, which they build up during their pelagic phylosoma developmental phase, and will not feed again until they have completed each of the 4 Puris developmental stages and have successfully instarred morphed to agile juveniles. Planktonic Puri are naturally pelagic, meaning living in upper water levels of ocean seas, and nectonic, meaning able to swim and move independent of ocean currents. But as they continue to develop, they are now transitioning to benthic, meaning lacking in plant nutrient, becoming permanent seafloor dwellers. The forward vertical and lateral swimming ability of clear Puri declines as they continue to develop through the late Puri stages. This means they will eventually choose to crawl rather than swim. This can be a detriment if the Puri have to complete exceptionally long migrations from the more oligothropic depths, as they may be unable to reach the security of grass zones. This would make them far more susceptible to predators or the inability to find sufficient food sources. There are several theories on what mechanism is used by planktonic Puri that guide them from the edge of the continental shelf to the safety of the nursery grounds near the shorelines. Underwater sounds is a theory that the planktonic Puri are able to navigate towards the coastlines, perhaps by the use of specialized setae on their antennae, allowing them to actually sense the vibration. sound waves caused by the waves breaking on the shores. Water chemistry is also plausible, as it is already recognized that crustaceans have highly developed chemosensory abilities, which have been shown to play a key role in the settlement behavior of species such as crabs. This suggests that subtle changes in water chemistry may be instrumental in triggering the metamorphosis in the final stage phylosoma, and perhaps a scent, which is secreted in the urine of settled lobster populations, provides some guide to subsequent inshore movement. Other possible theories are the ability to utilize magnetic fields, celestial cues, electrosense, hydrodynamic cues, and behavioral observation, and currently science yet fully understands what actually attracts these little sea bugs on their long shoreward migration. Then it is plausible that all of these theories are correct and are utilized in combination to guide them towards the shallows. Whatever cues triggers these planktonic Puri, they leave their pelagic zooplankton nursery and begin this life and death migration. For them to even have a chance to reach maturity, they must first successfully reach the sanctuary of coastal nursery grounds. Here in the Danagat Islands, Saragao Islands, and Siargao Island areas, we have the perfect seafloor topography to provide the natural cues to trigger both instar and Puri settlement. We are in very close proximity to the extreme depths of the Philippine Trench. The outer edge of the continental shelf runs the length of the trench. There are plenty of small, unpolluted, protected bays and estuaries with lush seaweed, microalgae, and coral zones, which are the natural nursery grounds for post-phylosoma Puri and early-stage juveniles. This has helped develop a healthy, thriving lobster grow-out aquacultural industry in this region. Puri are the heart of lobster aquaculture, and availability dictates the long-term viability of the industry. Side note: the 4 distinctive stages of Puri development, planktonic, clear, H-phase, and pigment, are each widely cited as non-feeding phases. I personally believe that this may be a little misleading, as experienced grow-out lobster farmers and multiple credible research feed studies came to a different conclusion. These lobster farmers and feed study researchers routinely sort their Puri by individual size and stage of their development. They then isolate them together in enclosures and completely separated from agile juveniles. They maintain strict observation of their Puri and still report measurable mortality rates due to active cannibalism. The 2 highest mortality factors cited in these studies and the lobster farmers is active cannibalism and the inability to free themselves from their older exoskeleton, referred to as morph death syndrome, NDS. My personal belief is to err towards caution and believe growers should provide sufficient mesh bundle shelters for their Puri as they are developing in your grow-out nets and cages, especially during the final pigment Puri stage of development. Also, remember that spiny lobsters are poikiliotherms, so maintaining optimum water temperatures and salinity levels for Puri is an important factor to successfully maximize their growth development. Planktonic Puri stage. Stage 1 post-larval Puri stage, lasting 2 to 4 weeks. The plural term is Puri, and the singular term is Panulirus, meaning little boy, Greek. The average size of planktonic Puri has a total body length, TL, of 30 to 35 millimeters, carapace length, CL, of 5 to 9 millimeter, and weighs less than 1 gram dry weight, TW. Currently, much of this nectonic phase, Puri, is still a mystery for researchers, as this occurs on the edge of the continental shelf at distances up to 1,500 kilometers from the shorelines. In the wild, 10% of Phyllosomatas survive the penultimate instar, morphing to the planktonic Puri developmental stage. For the first 24 hours after the penultimate instar molt, planktonic Puri are fragile, soft-bodied, and are not very active, but 24 hours after the penultimate instar, planktonic Puri are highly mobile, possessing both strong vertical and lateral swimming ability, and possesses a swimming speed of 7 kilometers, 4.3 miles per day. After the penultimate instar, planktonic Puris begin their long migration from the edge of the continental shelf to the shallow grass and coral zones of the shorelines. They generally swim during the new moon, no moon, lunar phase, which lasts 2 to 3 nights monthly, but perhaps this extends up to 5 to 6 nights per month if they also swim during the darker crescent moon phases. Being nocturnal, they shelter during daylight hours. They can complete this migration distance in 14 to 26 days, which shows that the current greatly assists these Puri to increase their swimming speeds. During this migration, these planktonic Puri are also facing a ticking physiological time clock. Planktonic Puri are naturally pelagic and nyctonic, but physiologically they are transitioning to becoming benthic seafloor dwellers. The 4 developmental stages and 3 physiological phases of Puri. This has been the area of my research which has been the most confusing, conflicting, and aggravating. There is simply too little written on the actual 4 developmental stages and 3 transitional phases of Puri. Most sources only reference 3 developmental stages, clear, H-phase, and pigment. But I personally believe there are, in fact, 4 and perhaps even a 3 to 4 day long 5th stage. Researchers have documented live capture post-molt planktonic Puri having been collected at distances of 200 to 500 kilometers out beyond the shelf break of continental shelves. So these researchers sufficiently validated the belief that the natural penultimate molt of latent stage 11 philosoma generally occurs 100 to 500 kilometers beyond the shelf break of the continental shelf. And as mentioned earlier, latent stage 11, 10, and perhaps 9 philosoma prematurely carried into areas with a water depth less than 200 meters appear to be cued due to quick water pressure increases. During my own personal research on the subject of the Puri developmental stages, I have actually found very little written on the subject, certainly nothing which actually documents the developmental stages and the individual transitional phases of Puri. Just how long is each of the Puri developmental stages? How long is each physiological transitional phase? I have read researchers citing the groundbreaking, successful consecutive raising of Panulirus ornatus philosoma from egg stage through penultimate instar molt. I have read a researcher cite a groundbreaking, successful mating of a second-generation Panulirus ornatus female. But as of yet I have not discovered any documentation on the individual Puris developmental stages or phases. So at this point, the information which I can share on this subject is limited and sketchy at best. What I have written here on the subject is an amalgamation of researchers' tidbits and knowledge gained from local Pueri juvenile fishermen and local lobster farmers. So if there are inconsistencies, I apologize, but know that I have tried my best to be factual. It has been this very issue which led me down the path years ago and eventually led me to start building this small micro hatchery. And God willing, if my little hatchery here on the edge of the mangroves proves successful, which it should, I promise that I will thoroughly document the Puris developmental stages of the Panulirus ornatus and then openly disclose all things relevant to the subject. Researchers primarily choose to just gloss over the subject, which is a real shame as it is so relevant and important to the lobster grow-out aquacultural industry. Clear post-molt stage. Stage 2 postlarval Puri phase, lasting 2 to 3 weeks dependent on water temperature and water salinity. During the long migrational swim, these Stage 1 postlarval planktonic Puri face an inevitable physiological transition from free-swimming translucent nectonic Puri to later becoming permanent benthic seafloor-dwelling individuals. These free-swimming translucent planktonic Puri will eventually choose settlement on the ocean floor. Once this settlement occurs, there is a physiological cue triggering the first of 3 non-instar, non-molt Puri physiological transitional development phases. This first physiological transition begins 3 to 4 days after this seafloor settlement, which is a Puri color transition. Their translucent planktonic Puri physiology will quickly turn to a milky coloration. The beginning of this milky color transformation signifies the beginning of their Stage 2 postlarval clear planktonic developmental stage. This is the first of 3 non-instar, non-molt physiological transition phases of Puri development. There are in fact 3 such non-instar, non-molt physiological transformations as Puri progress through all 4 Puri developmental stages. These transitions are the Stage 1 planktonic Puri to Stage 2 clear Puri, the Stage 2 clear Puri to Stage 3 H-phase Puri, and the Stage 3 H-phase Puri to Stage 4 pigment Puri developmental stage. At the conclusion of this latent Stage 4 pigment Puri developmental stage, they will Puerulus instar, molt, transitioning to Stage 1 agile juveniles. And yes, I see it a bit odd that translucent clear Puri are referred to as planktonic, whereas those of milky color are referred to as clear Puri. I don't make up the rules, rather I merely follow them myself. It should be noted that the forward swimming ability of clear Puri will steadily decline as they further develop through their clear Puri stage of development, as they transition from free-swimming nectonic individuals to permanent benthic seafloor dwellers. Note, during the clear H-phase and early pigment stage of development, the Puri has a noticeable flattening of their carapace and a noticeable ridging extending the length of both sides of their carapace length. H-phase stage. Stage 3 postlarval Puri phase, lasting 2 to 3 weeks depending on water temperature and water salinity. As clear Puri enter their second non-instar, non-molt physiological transition phase, their smooth milky monocolor coloration begins to display darkened pigmentation of their exoskeleton. This signifies the beginning of the Stage 3 H-phase Puri developmental stage. As these H-phase Puri further develop, this pigmentation will make more distinctive markings more prominent. H-phase Puris are losing their forward swimming ability and will soon be exclusively benthic seafloor-dwelling individuals. Note, during the clear H-phase and early pigment stage of development, the Puri have a noticeable flattening of their carapace and a noticeable ridging extending the length of both sides of their carapace length. Pigment stage. Stage 4 postlarval Puri phase, lasting 2 to 3 weeks depending on water temperature and salinity levels. As H-phase Puri enter their third non-instar, non-molt physiological transition phase, their pigmentation color becomes even more pronounced as their exoskeleton further darken, displaying their true distinctive markings. Now fully pigmented and darkened in distinctive colors, these pigment Puri now for the first time resemble little facsimiles of their parents. During the earlier Pueruli stages, so many of the 22 Panulirus species look so similar that researchers and experts must wait until the pigment Pueruli stage to properly identify the individuals. These pigment Puri are now exclusively crawling on the seafloor, having lost their forward swimming ability. They are now true benthic seafloor dwellers. Note: once the Puri transition to latent pigment Puri stage of development, their once noticeable flattened carapace shape and side ridging of their exoskeleton now show a rounding and a noticeable smoothing of their carapace. This rounding of the carapace exoskeleton is a visual marker for Pueri juvenile fishermen and lobster farmers to know the nearing of their Puerulus instar. This allows both lobster farmers and lobster fishermen with grow-out or nursery nets and cages to know when to properly isolate these individuals and provide sufficient shelter. Once they have further developed to latent pigment Puri, they will face a difficult instar molt, which historically has a 40% to 60% mortality rate. Individuals which successfully complete their Puerulus instar molt transition to Stage 1 post-Puri agile juvenile developmental stage. Agile juveniles historically have a higher than 90% survival rate, reaching the 500-gram buyer's market weight. Post-Puerulus juvenile developmental stages. These are 4 distinctive post-Puerulus juvenile developmental phases, which are referred to as the agile stage, post-agile stage, late juvenile stage, and sub-adult stage. The 4 post-Pueri stages are their fastest physiological growth phases. During these 4 juvenile developmental stages, they will quickly grow from a carapace length CL of 3 centimeters and a weight of 3 grams dry weight to a carapace length CL of 10 to 15 centimeters and a weight of 800 grams dry weight. Agile and early developed post-agile juveniles have a natural tendency to be solitary by nature, but as post-juveniles continue to further develop, they become more communicable. Juveniles are also aggressive and cannibalistic. Sufficient shelter should be provided within their grow-out nets and cages. Research studies have showed that when deprived of shelter, juveniles actually feed less, grow more slowly, and have considerably higher mortality rates due to injuries and acts of cannibalism, especially during instar molt cycles. This is an important fact to consider when establishing husbandry protocols for your grow-out aquaculture setup. Providing shelters at all stages of juvenile development reduces environmental stresses, which assist their immune systems. Researchers have proven that when sufficient shelters are provided, juveniles have a measurable increase of food intake, allowing them to gain weight faster and develop quicker. Researchers conducting feed studies on juveniles at each of the 4 juvenile developmental stages have shown that when individuals are fed diets that have insufficient nutritional value or are administered in inadequate feeding ratios, result in a significant increase of acts of aggression and cannibalism, especially during instar molt cycles when individuals are most vulnerable from acts of aggression from other members held within their enclosure. Cannibalism increases in enclosures housing individuals of mixed sizes. Individuals are more vulnerable to acts of cannibalism during instar molt cycles. Once individuals complete their instar molting, they are still vulnerable to aggressive acts for up to 8 to 10 days if housed with larger, more developed lobsters. This is primarily due to the fact that the new molt exoskeleton is extremely soft right after their molt. In fact, for the first 10 hours after a molt, the new exoskeleton of a juvenile is so soft that it is not yet rigid enough to sufficiently support their own weight to properly stand. Depending on the nutritional quality of the diet provided to the individual, it could take as long as 8 to 10 days for their new exoskeleton to fully harden. It is for this reason it's best to regularly separate and individually house juveniles by development size. The new exoskeleton for a newly molted juvenile can harden enough in 3 to 4 days to protect the individual from acts of aggression by individuals of the same size juveniles. But larger juveniles, subadults and adults, have larger and more developed mandibles. It is for this reason that housing individuals of different developmental sizes in single enclosures, that young juvenile individuals during instar molt cycles need to be removed and isolated for a period of 8 to 10 days before being reintroduced back into the housed population. Studies have shown that Panulirus ornatus juveniles grow on an average of 4 mm carapace length CL per month, and that their growth rate to juvenile is directly affected by the water temperature. One study conducted on Panulirus ornatus juveniles found that juveniles maintained at 30 degrees Celsius actually grew 31% faster compared to those kept at 26 degrees Celsius. This is due to the shorter molt intervals in large juveniles maintained at 30 degrees Celsius water temperature. But studies also show that aggression and cannibalism increases as the water temperature increases. Agile juvenile. Stage 1 PostPuri juvenile. This is the first post-larval feeding phase. No longer nectonic, the agile juveniles are now fully benthic, meaning bottom-dwelling. Agile juveniles are nocturnal, solitary, and both aggressive-natured and cannibalistic. The agile juveniles have a carapace length CL of 4 to 6 mm and weight of 3 to 7 grams dry weight, are certainly more desired than Puri-sized individuals. This is because Puri typically experience 40 to 60% mortality during the instar, molting them to the agile juvenile stage. Puri also can experience even higher mortality rates due to their young, fragile physiology. If Puri are subject to mishandling, adverse environmental stresses, exceptionally long travel distances, they can actually experience shock. And if this happens, then mortality rates of delivered Puri can be as high as 80% to 100%. This is why agile juveniles weighing 5 to 10 grams dry weight are more desired by lobster farmers. Agile juveniles have had time to rest after settlement and have further developed, making them larger and stronger, and fare much better when being transported over long distances. If agile juveniles were delivered to the same lobster farmer, he could expect to generally experience survival rates of 90% or above. There is a new type of aquacultural business developing within the industry, which are best termed as nursery grow-out ventures. They are both fishermen and buyers who now place the Puri in grow-out nets and cages to house them for an additional 3 to 6 weeks. Once the Puri have instarred to agile juvenile, they will house them for a minimum of 2 additional weeks, which allows them sufficient time to feed once again. This practice allows these early stage agile juveniles to begin to replenish their depleted reserves, allowing them to strengthen and gain weight before selling or transporting these individuals. Post-agile juvenile. Stage 2 PostPuri juvenile. Nocturnal, solitary, benthic bottom-dwelling, aggressive and cannibalistic. Juvenile lobsters are solitary by nature until they develop a carapace length CL of 35 mm, after which there is a characteristic shift and they become sociable. Late stage juvenile. Stage 3 PostPuri juvenile. They are nocturnal, solitary, benthic bottom-dwelling, aggressive-natured and cannibalistic. Generally, once latent juveniles reach a 10 cm carapace length CL and a weight of 170 grams dry weight, they are officially transitioned to the subadult developmental stage. Subadult. Stage 4 PostPuri juvenile. They are nocturnal, solitary, benthic, bottom-dwelling, feeding phase in both aggressive-natured and cannibalistic. Subadults typically have a minimum 10 centimeter carapace length (CL) and 170 grams dry weight. The first buyer's market weight is 500 grams. Once subadults reach a carapace length (CL) of 15 centimeters and a weight 800 grams dry weight, they have transitioned to the adult developmental stage. Adult, Stage 1, post-juvenile. They are nocturnal, solitary, benthic, bottom-dwelling, feeding phase, and they tend to become less in both aggressive behavior and cannibalistic. Adults have a minimum carapace length (CL) of 15 centimeters and a weight 800 grams dry weight. Most grow-out operations sell stock at the 500-gram buyer's weight while still subadults, though they are growers who house adult individuals 800-gram plus will further house, feed, and care for individuals to sell at the higher 1,000-gram plus size, second buyer's market price. These growers house the individuals until the lobster fishing off-season, when they then bring a higher price per individual. Generally, when a Panulirus ornatus exceeds a carapace length (CL) of 90 mm and a weight of 1.2 to 1.4 kilogram DL dry weight, they are then sexually mature and capable of successfully breeding. Females can mature and successfully breed within only 2 years from the hatchling neplosoma stage. This is considerably faster than most species of lobsters. During their first spawning season, she can breed 3 to 4 times and produce 800,000 to 1 million viable eggs. The female can easily carry 200,000 to 600,000 viable eggs, but I've read that larger, more mature Panulirus ornatus females can carry over 8,000 viable eggs. The reproductive success of the male is relative to the size relation to that of the prospective mate. Newly settled Puri will remain among the shallow grass and reef zones, where they continue to develop for approximately 2.5 years. Once they have reached the carapace length (CL) of 95 mm, they will then leave those shallows and seek deeper waters, where they will spend the rest of their adult life. When held captive, the Panulirus ornatus has fastest growth rate and grows larger than any of the 22 Panulirus species. They also adapt well when held captive in tanks for extended periods of time. In fact, I have seen it written where they are successfully captive for periods of 7 to 9 years. It is possible that individuals have been held captive for longer periods of time, but I just have not personally seen anything written citing longer captive times. Adult Panulirus ornatus can only increase in size when they instar molt, and though the growth of internal tissues continues, their growth size between instar molts does not. As these adults continue to mature, the increments of the instar molts will decrease, while the time intervals between the instar molts will increase. This cause and effect cycle means that their growth rate actually slows as they continue to grow in size. Here is another contradiction, which is the maximum reported total body length of 60 centimeters, 25 inches. The previous record-holding lobster weighing 28 pounds, which was caught on the coastal city of Wingling, China, and sold at a fish market there. So I assume this was a benchmark used to determine the weight and dimension of the species, as any other assumption would merely be conjecture at best. But recently a new recorded size behemoth Panulirus ornatus, weighing 25.4 kilograms, 56 pounds, was caught and sold for over $95,000 at auction. So logic dictates that their total body length is actually longer than the current belief of being 60 centimeters, 25 inches. Now, if this doubling of total body mass between the former and current record holder also means that the total years of their individual lifespan must now be reconsidered, I simply cannot provide any useful factual information relating to that topic at this time. But I suspect some revision may be necessary. Until then, the current belief is that the lifespan of Panulirus ornatus males is upwards of 35 years, and the females can live upwards of 50 years. Non-feeding and cannibalistic phases. Nepulous. Egg hatching, first 2 to 3 hours before first instar to stage 1 Philosomata. Non-feeding. Philosomata. All 11 developmental stages. Highly aggressive, highly cannibalistic. Puri. Planktonic stage, clear stage, H-phase stage, and pigment stage. Non-feeding. Agile juvenile. Extremely cannibalistic until they reach 7 to 10 gram dry weight, where sufficient nutrition at proper feeding ratios and adequate shelter can help reduce acts of aggression and cannibalism. Highly aggressive, highly cannibalistic. Post-juvenile, primarily issued during instar, mainly due to insufficient diet or lack of adequate shelters being provided. Aggressive, cannibalistic. Late juvenile, primarily an issue during instar, provided insufficient diet or lack of adequate shelters being provided. Aggressive, cannibalistic. Subadult, primarily an issue during instar, provided insufficient diet or lack of adequate shelters being provided. Aggressive, less cannibalistic. Adult, primarily an issue during instar, provided insufficient diet or lack of adequate shelter being provided. Aggressive, less cannibalistic.